18E is the first 8-color STR system that has been developed for the European market, and it follows all the guidelines for recommendations of the loci to be included that is recommended by European National Forensic Science Institutes. The labs typically get a lot of challenging samples. We hear about mass graves being unearthed in different parts of the world, and these human remains have been subjected to so much of environmental stress, and they are clearly not in the pristine condition. So unlike DNA that you would extract from human blood, from fresh human blood where the DNA is of a high molecular weight and really good condition, these samples unfortunately are not in a very good shape at all. With the 18E, we have made sure that all the 16 recommended loci by the ENFSI are within 325 base pairs. So even if the DNA is highly fragmented, we have a much higher chance of getting a full profile because our primers are kind of sitting closer to each other. And this is possible only because we have an 8-color system. As of now, 18E would be the first 8-die STR kit that would be compatible with the high-throughput spectrum CE system and the low-throughput spectrum compact system. So we have 2 systems that would fit the different throughput needs of the lab, and both would be capable of running 18E chemistry.